Hello, everyone. First, I'd like to really thank the committee for choosing me, my nominators for nominating me, all the mentors and students and collaborators and participants who've participated in this work. I have a slide at the end thanking everyone, but I just wanted to get that off my chest at the beginning. But now I can also talk to you about a little bit about science. It would help if I take out my clicker. Great. So what can we learn about cognitive neuroscience from people born with sensory or motor losses? So the question, you can do a lot of different things in terms of cognitive insight and research with people who are born blind, deaf, or without hands. Specifically, I'm looking into what drives brain organization. And this amazing ability of the brain to develop into functional, complex, anatomical substrates of what our minds can do is a pretty fascinating phenomenon. And we all know that it is driven by both nature and nurture. We're not going to try and say or, right? We know that there's an effect of environmental. This is way too strong. I think this is meant for taller people. We all know that there's an effect of environmental experiences on brain development. We also know that monozygotic twins have more similar brain patterns as compared to dizygotic twins. And working with and studying the brains of people born blind, deaf, or without hands allows us to test what all those deprived cortices bits do in the absence of that fairly large, meaningful experience of the environment. Specifically, we're going to look today at the dependence on sensory and motor experience, the potential and extent of plasticity, how much plasticity is there in the human brain, and I'll argue also about the representation level present in brain regions, both in these populations and in the typically developed brain. Now, I'll walk you through obviously too many studies in people born blind and people born without hands. And I'll start with some old work in blindness as it sets up the picture and the theoretical concepts of what we're doing now. So for a very long time, people have been looking into what does the visual cortex do in people born blind? What does it do without vision? And I've somewhat contributed to this work, working with something called sensory substitution, whereby we taught people born blind how to see using sounds. We used a really simple translation, which allows you to topographically translate images into sounds. I won't bore you with the details. I'll just give you a small demonstration that will make it clear. You have a link between the frequency change over time, in this case it's a dropping tone, and the visual representation. So a diagonal will be represented by a dropping tone or a rising tone. And you can combine different features in Audition to create complex shapes. The point is that you can topographically map any image into a sound, allowing people to perceive complex visual images using their intact senses, even if they're blind. And that allowed us to take people who are born blind, put them in the scanner, and look into what their visual cortex is doing as they perceive things that would normally elicit the dissociation between the ventral and dorsal stream. We know that we have a ventral what and dorsal where streams, and we were able to replicate that with people born blind who were perceiving sounds that represents images as they were attending either to the shape or to the location of the simple stimulus in front of them. Similarly, we were able to train them to identify more complex images, such as images of faces and houses and letters and objects and body shapes and all of that, and show that they can actually understand these images, and then explore category selectivity in the visual cortex of people born blind. Now, again, just one example of a study showing this. We've shown a selectivity in the visual word form area, which is an area that's specifically selective for letters over other visual categories. This is in the sighted brain. This is where it's supposed to be. This is where we find it in the blind brain as they perceive sounds that reflect letters. In some more follow-up work on this, we actually showed that this area was only activated by soundscapes or by sensory substitution when they were identifying these shapes as graphemes, saying something more about the computational role of this region. But over the years, there's been a beautiful amount of work done by many, many people, some of which are here today, showing that the organization of the association visual cortex is not critically dependent on visual experience. In other words, these areas of the visual cortex can arise into a similar functional role in blindness. And that suggests, and has been called metamodal organization, that these regions develop those functions regardless of those low-level features and regardless of experience. In a way, that's a way of showing that these regions can abstract away from those low-level visual properties and perceive information and perform those computations regardless of the sensory modality. And it's also been shown that it happens in sighted people as well. So we're not saying something is uniquely plastically reorganized in the blind brain in that sense. These are areas that are multisensory in nature and can show this more robust role in the other sensory modalities in blindness. So why am I showing you all of this, right? Old works. I'm showing you this because in my lab, one of the things we're doing is trying to see whether this is a general principle of brain organization and plasticity patterns, and specifically looking into the motor domain. Now, shifting gears to the motor domain. In the brain, we have a lot of regions that are dedicated to different parts of our body. In light green here, you can see all the areas that have a preference for movement of the hands as opposed to other body parts. And we also know that some of those hand regions have a preference not just for twitches of the hand, but for performing specific types of actions with our hands, such as reaching or grasping. And there's even been a proposal by Michael Graziano that different areas in the motor system are actually involved in ethologically complex, complete actions. So what we're trying to do with this model, with extending this question into the motor domain, is see what is the core organizational principle. Are these areas dedicated hand areas that are specific to the somatotopic organization and are specific to the body part, or are they organized by those types of motor tasks? In other words, are those association motor regions able to abstract away from the low-level properties in the motor domain in the same way that the association visual cortex abstracts away from the low-level visual properties in vision, in blindness? Still following me? Awesome. So these are the types of people that I study in order to try and address these questions. I study people born bilaterally without hands. They are born with very little, if any, hand and arm residues. Now it stops when I do this. And that allows us to explore whether motor experience is actually crucial for hand-related brain functions to develop in their proper places, or to what extent the organization is specific to the hand or specific to the action performed with the hand. Now, I want to highlight that this population is a very small cohort, and these are all essentially designed as series of case studies. I'm very happy to talk and answer questions about why or how we dare make assumptions about cortical organization using small, unique cohorts. But for the moment, I'll just say that we are testing for the consistency across people who are typically developed and people born without hands. Because what we would like to show is that those regions are organized by the action type, regardless of whether I perform an action with my hand, I perform the same action with my foot or my mouth. I will not demonstrate right now. But if I were to do this and show this activation and organization in the brain, people could say, oh, when I'm using my foot, I'm just imagining hand movements and translating that somehow. If I do that with only people born without hands, well, they obviously cannot imagine hand use, but it could just be plasticity in these populations. By trying to combine data from both these populations in both these cases, we're trying to say something more meaningful about the ability of these regions to abstract away. Okay, so now it's showing it again. So in one of my works with Alfonso Caramazza, my postdoc mentor, we looked into this organization in people born without hands. We had Typically developed people and people born without hands perform reaching and grasping actions. As we said, this is one of the divisions of labor known within the hand regions. Obviously, controls did it with their hands. Individuals with dysplasia born without hands did it with their feet. And we can see a lot of regions highlighted in orange here that show this main effect of action, that care about what action is being performed, regardless of what body part is performing it. And of course, we can also decode within areas that are selective for hand actions in people born without hands what action they're performing. Together, this suggests that some of the association motor system may be organized for action types and not just body parts. I want to make sure that I'm not trying to say that these organization principles are the only organization principles, right? We expect there to be some mixed selectivity. We expect them to be some combination of those signals. Now, this is still very much in line with the Graziano model of these ethologically actions that are organized in the brain. We wanted to take this one step further and see whether that is limited to actions and brain regions that we have inherited from our primate ancestry. Or in other words, can we extend this principle into actions that are unique to humans, like tool use and writing? So I can show you one preliminary data point, and we'll have a lot hopefully coming out in the next few years. We're looking into tool use and decoding between the actions of tool use and grasping in training the classifier on hand trials and testing it on foot trials of the same participant. And we bring out the entire association motor system, including premotor cortex, superior parietal lobe, the cerebellum as well, unfortunately not shown here, but very much present. And within those regions, we can also decode not just the action performed by the hand in our controls, but also actions performed by the foot in individuals born without hands. Together, these data points, and I hope to have more in the future, suggest that the association motor system is organized also by action, regardless of the effector, regardless of the acting body part, representing this type of motor abstraction. And this abstraction is what can allow us to all do things like motor transfer, imitate an action that we see with one body part to another body part, write with our toes in the sand, and so forth. And that is also how we typically control our actions in daily life, right? When I reach out for this clicker, I guess, I never have to consciously think about how wide I want my digits to be, right? I have this more abstract action goal, and that is cascading down the action or the motor hierarchy to produce a complex action. And we think that this is one of the things that would allow us in the future to also potentially control prosthetics in a more meaningful way if we could harness these types of action representations. Now, we're testing all of these ideas across more actions, across observation and imitation, to see how far we can push this idea of abstraction and action type preferences. But overall, the reason I showed you both the blindness old work and this relatively new work is to show the principle that the association cortices across vision and motor function, and there's been beautiful works in deafness as well, across sign language, and Steve Lumber's works in cooling cat's cortex, showing that these areas process information typically from multiple sensory motor channels, and that allows plasticity for the same computations, even if you're deprived of one of those modalities. And that again shows us these regions are independent from sensory motor experience, abstracted from low-level features, and that's all fairly consistent. And now, the next question is, how far can we push this idea? What's going on in M1? Is M1 also abstracting away? Is M1 tied to the specifics, in which case you can expect a takeover by nearby body? If it's abstracted from the specifics, you can expect the area to be overtaken by the body part that functions as a hand, right? If you have a representation of fine motor control, our participants perform that with their foot, as you could see with Tisha carving the pumpkin. Alternatively, if this area is still tied to the low-level sensory motor specifics, you can expect it to be overtaken topographically, like what we see in people who lose their hands due to amputation later on. So we tested this, doing a simple somatotopic mapping experiment, and you can see the yellow area here in controls representing the preference for the hands. And you can see that in people born without hands, that area, outlined in white here, is completely overtaken by the adjacent body part, by the shoulder, by the abdomen, even though they don't use that functionally in their daily life to compensate. And this tells us that this region is not overtaken by foot movement, even though they were born without hands, even though they are performing foot actions since probably early childhood. These compensatory use and plasticity do not overcome the anatomical topographical constraints. Or we can phrase this as saying that the computation level of these regions cannot be abstracted away. It is tied to those sensory motor low-level processes. Now, that suggests that there's a gradient of abstraction here. And you can actually see that you have those pink areas, I should use this, right? You have those pink areas that are overtaken by the foot in other areas that aren't in M1. And you can kind of try to think whether we can use this type of reorganization or the pattern of reorganization as a marker for abstraction level in the sensory motor system. Whether we can highlight areas that are supposed to be hand areas, but are overtaken topographically in one color and say, okay, these are low-level areas, and highlight areas that are overtaken functionally and mark those as more abstract. And we're pursuing this idea to see whether we can use these types of functional markers. As I said before, we could also theoretically use this hierarchical gradient to think about how we can improve on prosthetic designs and brain-machine interfaces, which are currently very much driven by low-level parameters, which are harder and less natural and more effortful to control. Now, I've told you a lot about the consistencies between blindness and handlessness and deafness. So what about V1, right? We started off with blindness. What's going on over there? We were able in the past to show, using functional connectivity with Smadaro-Vardekaro, that if we look just at the functional connectivity from retinotopic seeds in V1, we can actually replicate a lot of the topographic functional connectivity patterns within the visual cortex. So there's a lot of intact organization that's going on in terms of the large-scale network architecture. But that should not surprise anyone in this audience, right? We have a lot of this connectivity wired up at birth, and it should not be affected, at least at this fairly large-scale level, by visual experience. Unfortunately, this only tells us something about the connectivity pattern, the network organization, and not about the function of V1. And when we think about the function of V1, that's where I'm stuck. Over the last decades, people have shown that the early visual cortex in blindness is responsible to anything and everything you can think of, or at least anything and everything that I could think of. They have shown that it is responsive to touch, to audition, to smell even, to echolocation, to language, to memory, to executive function, calculation. I am hoping I'm not missing anything important here. And both as a systems and as a cognitive neuroscientist, that leaves me stumped, okay? It either means that some of this is epiphenomenal, or it means that we are looking at something else. Either there's no meaningful functional organization, or there's no consistent functional organization. So, taking a leap of faith, I've tried to look into whether those results could actually arise from inter-subject variability. Whether at the group level, yes, we have meaningful increased activation for a lot of different things, but in fact, maybe each blind participant is using their visual cortex meaningfully for something else. And that highlights general questions of how variable brain plasticity may be, or more generally, how much is there a role of experience in generating the consistency that we've all come to love in how the brain is organized across individuals. So we've shown a few preliminary things, and I hope you'll bear with me. First of all, we wanted to see whether brain connectivity from early visual cortex in blindness is indeed more variable. And we've done that. There's huge differences between blind and sighted individuals in how variable their functional connectivity to the rest of the brain is. And these differences all stem from blind people showing more meaningful variability, more individual differences in how it connects to other parts of the brain. Now, more interestingly, these areas, the areas that are variable between blind participants, are also the areas that show functional connectivity plasticity. These are the areas that change their connectivity with other parts of the brain in the blind. And it looks like plasticity really generates variable outcomes. What are those variable outcomes? We can do some very simple hierarchical clustering to see what those profiles look like. And these are going to be fairly small, but maybe you'll believe me. You can highlight differences in functional connectivity from the visual cortex to the auditory cortex across different individuals, to the sensorimotor cortex across different individuals, and to the frontal lobe across different individuals, suggesting that maybe there are different functional developmental pathways for this deprived cortex. Now, these differences do not seem to stem from the etiology of blindness, from the cause of blindness, or for how blind they are, because this is a fairly homogeneous group. And it doesn't seem to stem from genetic causes or genetic diversity either, because these two nodes that I've highlighted here are actually two sisters that are blind from the same genetic cause, and they end up with fairly different brain pattern of plasticity. Now we're diving way more deeply into what those variable outcomes may be, how they're affected by compensatory experiences, and whether they carry behavioral advantages. And if they do, it is possible that they would be useful for tailoring rehabilitation and giving each person the assistive technology that may be a better fit to them. Now, obviously, there's a long way to go in order to show these things. This requires first showing that these individual differences are stable over time. Otherwise, we wouldn't dare use them to predict outcomes or to ascribe rehabilitation. And we obviously need to show their relevance to rehabilitation. And I'm well aware that extraordinary claims require extraordinary evidence, and we're not there yet, but I will show you 2 data points supporting each one of those ideas. First, and this is work done by Lenya Amaral in my lab. Keep an eye on her. She'll be going on the job market in a year or 2. We've looked into the consistency of individual patterns of functional connectivity in congenitally blind individuals that were scanned several times. These are people that I've scanned during my Ph.D., and they were scanned over 2 years in 3 different scans, including both task scans and rest scans. And we were able to train a classifier on identity of the participant on 2 different scans and test it on the 3rd, mixing those up, of course, with a really high and significant decoding accuracy, and with so many regions of the brain allowing us and having a pattern of different individual patterns, allowing us to decode identity of these individuals. More interestingly, at least for me, the patterns of plasticity, or the differences between each blind brain's pattern and the cohort of sighted participants, also remain stable over time, suggesting that they have stable patterns of differences and stable patterns of plasticity. Now, are there implications for this type of variability? And here I am fortunate to have the collaboration of Pavan Sinha and his team, who have a beautiful humanitarian and scientific project called Project Prakash, where they do sight restoration operations and treatment for kids, adolescents, and young adults in rural India. And what they see is that sight restoration following long-term early onset blindness, kids born with dense bilateral cataracts, is really non-trivial. A lot of kids do gain meaningful sight, but there's a really large unexplained variability in outcomes across individuals. And what we're trying to do together is look at whether brain connectivity before the operation can actually allow us to predict who would gain more sight, and whether that can be a determining factor in ascribing treatment. So again, preliminary results. But what we're looking at here is functional connectivity data prior to the operation, and whether that can predict the gain in acuity across those two time points, before and after the operation. We train a linear regression model on the preoperative data with cross-validation, and we find several regions that seem to show meaningful effects. Now, this is currently based on a somewhat small sample, again, 10 participants tested before and after site restoration. And we've pre-registered these predictions, and we're waiting for an out-of-cohort sample. But this does suggest that brain connectivity before the operation, again, if it pans out with a replication, may be meaningful enough to allow us to use that for rehabilitation, or at the very least, to tell us which networks are involved in functional improved sight. Now, over all of this, probably too much data, I hope I've shown that congenital blindness and handlessness result in functional plasticity. There's a lot of consistency in what goes on in the association cortices, right? They all retain the same function for other senses, other effectors. And this demonstrates that they are abstracting away from those low-level features, and by this I mean retinotopic location, muscle group, and the likes. The early sensorimotor cortex shows a different plasticity pattern in that it is specific. And together with the association cortex, handlessness seems to be a really nice model for looking at the cognitive hierarchy of action representations. Plasticity in early visual cortex and blindness, to me at least, is still unresolved, but it seems to generate more variable connectivity across individuals that's stable over time. We do have hints for similar individual differences in deafness as well. And those, and you can check out the preprint also by Lenya Amaral. And those are also linked to the time and delay in sign language acquisition. And I hope that in the future, maybe in a few more years at CNS, I'll be able to show you how individual differences in the brain may be informative to predict behavioral advantages, treatment outcomes, or some functional use. If you're interested in joining these lines of research, ping me. And I have so many people to thank, really. My mentors, Amir and Alfonso, my collaborators over the years, my current collaborators, my lab, and especially I want to highlight Yuqi Liu, who's already left my lab and is now in the Chinese Academy of Sciences in Shanghai, and Lenya Amaral, who's, again, look out for her. And I want to thank the Chen Institute for supporting this award. Thank my participants who have been in the scanner, as you could see, repeatedly and for long durations of time, and I couldn't have done that without them. And thank you all for listening.